Hello, everyone. Welcome back to ResearchWorks podcast here at EACD and IAACD in Heidelberg 2025. And with me now, we have another keynote speaker, Professor Jan Kirschner. Welcome to the podcast and to the conference.
Thank you. Happy to be here.
So lovely to have you here. Now, your keynote caught my interest, and it's going to be one that I'm sure people really want to know more about and come back to this episode. It is titled Gene Therapy for Childhood Onset Disability, Lessons from Neuromuscular Disorders.
Yeah, it's really fascinating to see how gene therapies nowadays really open a completely new avenue of treatments for disabled children and infants. And I mean, we have the experience with spinal muscular atrophy, and it's really fascinating how gene therapy has completely changed the course of the disease.
Yes.
And in particular, if we combine gene therapy for SMA with newborn screening, it's fantastic. So nowadays we see infants that previously were not even able to sit and required artificial ventilation and tube feeding.
Yeah.
They nowadays survive without respiratory support, without tube feeding, and they even start to walk.
Yeah.
So that's really fascinating.
It is absolutely amazing. We were just sharing with you before that at our clinic back at Healthy Strides, we're seeing more children with SMA who they're learning how to walk, and they are actively part of their community and living their life. And it's an incredible thing to see. How long has it taken for gene therapy in SMA to, I guess it's nice to know the history in terms of how it can come to a place where it's now in effect?
Yeah, so of course, it's a long history, starting with the detection of the genes, the genetic cause of SMA, which is the SMN1 gene. And then we realized that there is a second copy, the SMN2 gene, which can also be used for some treatments by splicing modification. So it's we could say it's like a bad copy of the gene, which can be influenced by splicing modifiers to produce more protein. But then after that, we realized that also an approach is to add the intact SMN1 gene by gene therapy. And we use viral vectors to transduce the cells. So basically, these viral vectors carry the genetic information that is missing in the neurons. And we basically infect infants with this viral vector. And then the viral vector would transport genetic information into the motor neurons. And then the neurons can start to produce the SMN protein, which is missing. And then motor function does improve.
Yeah, it's really, truly incredible. What has been learnt from, I suppose it's a good example, isn't it, SMA? Are there other examples or lessons that we have learnt from genetic therapy and the new discoveries?
Yeah, I mean, what we have learned also from SMA is that there is a therapeutic window. Right. So you have to treat early and ideally pre-symptomatically. So that's why we are doing newborn screening now. So you start treatment before the disease is manifesting. And we also see if you start treatment late because diagnosis was delayed or the patient is in advanced stage of disease, the gains you see are still there, but they can be minimal. So it's really important to identify the right time point and to treat early.
Yeah.
And unfortunately, not in all diseases, we have this therapeutic window. Sometimes infants with neurologic diseases are already severely affected at birth, and then it might be difficult to really to achieve meaningful improvements by adding genetic information.
Yeah, it's an interesting point, isn't it? I mean, increasingly, what I've also heard at this conference as well, and just doing a bit more reading, is the genetic background for cerebral palsy, for example, and understanding what that might look like. And I remember when I was being trained, we always said that cerebral palsy was did not have a genetic cause. But we know now with more research that that's not necessarily the case.
Yeah, that's right. So on the one hand, there are genetic disorders, monogenetic disorders, which can mimic cerebral palsy. So it's important to differentiate this from classical cerebral palsy. Yeah. But there are also genetic factors predisposing to cerebral palsy. So at the moment with gene therapy, we are focusing on monogenetic diseases.
Yes, okay.
But of course, in the future is also, we can think to modify genetic factors that influence the course of the disease. So there's potentially also a field in diseases which are not monogenetic like cerebral palsy. But this is far long way to go, I think.
Yeah. We did speak with Darcy Fellings the other day, and she was saying it's really quite at the beginning phase at the moment for cerebral palsy. Another area I know that you're really interested about is Duchenne's muscular dystrophy and what's happening in that space at the moment. Can you fill us in on what's happening?
Yeah, so for Duchenne muscular dystrophy, we face several problems also compared to SMA. So as I said before, we use the viral vector to transport the genetic information. The problem is that the packaging capacity of the vector is limited.
Right.
And the dystrophin gene is one of the largest genes we know. And it's not possible to package this into a single adenoviral vector. So we have to reduce the gene size. So we cannot introduce a complete dystrophin gene. We have to use what we call a microdystrophin. So we make a small gene product that can be introduced into the cells. But of course, this microdystrophin has not the same function as the full-length protein. So there is some limitation concerning the gene size.
Right.
And also the target tissue, which is muscle and not neurons, is different. And there is turnover of muscle cells, and then the gene might dilute out after a longer period of time. So we don't know how long the effect might last. So what we have seen in clinical trials, we see some benefit, but it was not so convincing. And clinical trials were also negative concerning the primary outcome.
Okay.
But nevertheless, of course, there's a huge unmet need in this disease. So the FDA has decided to approve the drug despite a negative phase 3 trial. And in Europe, we are still waiting for the decision. And we also have seen side effects in gene therapy. We have seen this in SMA very rarely.
Yeah.
And we have also seen this in Duchenne muscular dystrophy. And actually, very recently, there were 2 fatalities related to liver failure. And so there are now concerns about the safety profile also. So I think what we really need, I mean, we cannot wait with approval to test a therapy in 1,000 patients because it's a rare disease and it's impossible. This would take 20 years.
Of course.
But I think what is really keen that we continuously monitor effectiveness and safety. We collect real-world data to really establish a learning system so we can adapt the use of the treatment. We can adapt our immunosuppression treatment, which might be needed to avoid side effects. So I think it has to be like, I would call it a learning system rather than a one-time approval and then you can use it. You have to continuously collect information and update the community about safety and effectiveness.
It comes down to methodology, doesn't it? So the more traditional methodological processes are not supportive for this particular population. You say learning system. Can you tell me a bit more about that? That's an area of great interest for me.
Yeah, I mean, absolutely. I mean, in common diseases, typically you would test a drug in a phase 3 study with thousands of patients before you approve the drug. But as I said, this is not feasible in rare diseases.
That's right.
And we cannot further delay approval for these drugs because they're really needed. But I think what we need is to systematically collect real-world data in well-established registries which are internationally aligned.
Right.
And then we can identify new safety signals. And also in SMA, I mean, we have identified severe adverse events like thrombotic microangiopathy, which is very rare, probably less than 1%.
Right.
But this has not been observed in any clinical trial, only when thousands of patients were treated after approval.
I see.
And I think this is what we really have learned, that this continuous collection of evidence is really important. And some treatments might even disappear from the market because at the end it turns out that the benefit-risk ratio is not really worthwhile.
Yeah.
And others, it might really be confirmed and they are used even in other populations, in older infants and children.
What does that look like in reality? As you talk about this continued collection of data in registries, is there like a combined resource of data, or is it through summits where there's meetings? Like, what does that look like practically?
Yeah, so of course it depends a lot on the health system. So it's not so easy to use uniform registries on an international level.
Yeah, sure.
But at least you should align what kind of outcome measures you are collecting. And for example, in Germany, we established a registry which is called SmartCare. And HTA agencies in Germany, they made it mandatory to collect data of treated patients in this registry. So now we have almost full coverage. We have more than 2,000 SMA patients registered in Germany. And we have already addressed safety and effectiveness of gene therapy for SMA in a cohort of more than 400 patients. So I think it should really be mandatory to collect real-world data from these patients because if you invest a lot of money, a lot of effort, you should really know what happens to the patients after 5 years, after 10 years. And I think it's irresponsible to use this kind of treatment without ensuring follow-up to learn from the experience and to develop the evidence.
Sure. Is there a particular case that comes to mind? I often think about, when there's more, I suppose, rare conditions, you've got methodologies like single case designs, you've just spoken about registries. I guess what does the future hold for this area based on what has been learnt before in terms of how we can better do research in this space?
So I think what is really key in rare diseases is networking.
Okay.
Because a single center, you can never achieve what you can achieve in a network. So it's really important to collaborate, to establish data collection together, to share information. Also, timely sharing of safety issues is very important.
Yeah.
If you use the official pharmacovigilance pathways, it often takes months or even years that you get aware of a severe adverse event at another center.
Yeah.
So I think we need networks to really share this information in a timely manner. I really want to know if there's a severe side effect in the US and I'm treating a patient next week. I want to be aware of this risk and inform families accordingly. So I think we need new systems also to share this information
almost in real
international
time.
networks.
Yeah, that's right. You can see how beneficial that would be. What's the area that you feel like is on the horizon at the moment based on what you have seen or you're working on that I suppose excites you the most?
So what is definitely interesting if we talk about the development of gene therapy, that at the moment we are using natural viral vectors.
Okay.
But there's now also a lot of engineering of vectors. So they are not naturally occurring viruses, but we use engineered vectors, which hopefully will have, for example, more packaging capacity. They might also be less provocative to the immune system, so cause less immune response. So this is a very interesting area that we now try to specifically develop vectors for gene therapy, which hopefully have a better tissue transduction and less side effects.
Yeah, well, that's very interesting. I guess that's the advancement of technology at the moment, isn't it? And it's keeping up with what's happening. Now, the main title that you hear was Lessons from Neuromuscular Disorders. If you had to bring it down to, I guess, your top 2 lessons that you want people to really capture and remember from this conference, from your keynote, what would they be?
So I think what is really key that gene therapy can be extremely successful, but that's not the same for all diseases, unfortunately. So there are some specific diseases where you have a therapeutic window, you have ideal target tissue like neurons, for example. It's easy to get your gene therapy to the target tissue if you don't have turnover of the tissue.
Yeah.
But in other diseases, it's more complex. So I think it's important that gene therapy is really promising, but it's not so easy. Sometimes you think like, well, you just take a vector, you introduce a gene, and then you would give an infusion, and then the patient is cured.
Yeah.
But that is definitely not the case. And gene therapy will never replace the current multidisciplinary treatment these infants and children need. And that's also important. Sometimes people feel like, okay, I can use gene therapy. I just provide the gene therapy. And they don't really take care of all the other aspects because most patients are still symptomatic and sometimes severely symptomatic. So we should not forget about multidisciplinary treatment for these diseases. It's still a cornerstone of treatment and will most likely never be replaced by gene therapy.
Yeah, absolutely. And everything you're saying here resonates with a lot of the topics of the conference at the moment. It is collaboration, developing networks is just so key. The value of early intervention and the value of actually working together in a multidisciplinary, you can never replace that. And at the end of the day, the children and the families we work with, they're complex networks and it requires a network of people to be able to support them. So, yeah, very exciting direction. I imagine it's a very rewarding and challenging area to work in. But I'm so grateful you were able to share this information with us into a level that I can understand very well. So thank you so much for joining us on the podcast today and for your keynote presentation.
Okay, thank you. It was a pleasure.
Oh, it was wonderful. Yeah. To all of our listeners, I hope you really enjoyed that. Very intriguing and an integral part of the future direction. So hope you enjoyed it. Stay tuned for so much more. Bye.
Thank you. Bye-bye.